Hi, I'm Dr. Pierce, and welcome to this video on biological membranes. So you've probably seen a diagram like this before, showing the basic structure of a biological membrane. But what I want to do in this video is not simply discuss the basic elements of membrane structure and function, but also show the connections between biological membranes and other key topics in biology. So it's going to be prudent first to have a think about what membranes actually do. So the first role that any membrane plays is to create a compartmentalized environment that is different on the inside than it is on the outside. So you can kind of liken this a little bit to a castle surrounded by a moat. And if you can imagine that moat allows the people inside the castle to control what comes in and what comes out of it, then you can also imagine that the inside of the castle is going to have a particular culture or environment to it. And that's exactly what cell membranes do. And this gets to our first connection to the big ideas of biology, that being homeostasis. So by controlling what crosses the membranes, cells or other organelles can maintain a specific, narrowly defined internal environment that allows it to carry out its necessary specific functions. So a good example of this are those little sacs of death we know as lysosomes. So with an internal pH of 5 and a charming menu of hydrolytic enzymes, these little beasts can carry out specific functions without actually harming the rest of the cell. And that's all because of the membrane and the way it helps create and maintain a unique internal environment. Another big idea comes from the fact when you take a look at the basic components of a membrane and have them interact to form a complex structure, you get new properties and functions that did not exist before. So the basic components of a membrane include phospholipids, and in the case of animal membranes, you've got cholesterol. You've got a whole myriad of proteins involved in these things, and you also have glycoproteins and glycolipids, which really just are phospholipids and proteins that have sugar attached to them, hence the prefix glyco. In and of themselves, these components have properties, have basic properties, but it's not until you bring them together that you actually form something pretty special, and that is the basic biological membrane. And these biological membranes follow a model called the fluid mosaic model, which we'll talk a little bit about now. So the fluid part comes from the structure of the phospholipids, which are amphipathic molecules having both nonpolar or hydrophobic fatty acid tails on the one end and hydrophilic and or polar heads made up of the phosphate group at the top. So if you take a whole bunch of these phospholipids and you force them into water, what's going to happen is the hydrophobic tails are going to try to avoid any water at all costs, and they will come together and form a phospholipid bilayer. Now this phospholipid bilayer has some unique properties. It's got a hydrophobic core due to the interactions of the hydrophobic tails, the fatty acid tails, and yet both surfaces are hydrophilic and interact with the aqueous solution on the outside of the cell as well as the aqueous solution on the inside of the cell. So as mentioned before, the lipid bilayer gives the membrane its fluid nature. So what does that really mean? Well, basically biological membranes are not rigid structures like the shell around an egg, but rather they have the property more like that of salad oil. And as you can see in this short clip from the excellent video called The Inner Life of the Cell, you can see that the lipid bilayer has this wave-like motion, this fluid-like motion, and the phospholipids in the bilayer are kind of shifting back and forth, and in fact there's a ton of lateral movement of these phospholipids in the membrane, but it's all being held together by those hydrophobic interactions. The fact that the membrane is a fluid structure is critical in understanding some of the key concepts in biology. So for example, fluidity is required in order for vesicles to bud off organelles like the Golgi and subsequently fuse with other structure like the plasma membrane. So the whole connectedness of the endomembrane system, from the rough endoplasmic reticulum to the Golgi and then on to lysosomes and the plasma membrane, is dependent on this fluid nature of the membrane and vesicle formation. Another kind of interesting aspect of this is your immune system, where phagocytes like this macrophage are used to engulf pathogens that are in your body. Again, this engulfing of a pathogen, which leads to phagocytosis and the destruction of the pathogen, is dependent on the macrophage, the macrophage, able to wrap itself around and fuse its membrane on the other side. So again, membrane fluidity is critical in that particular function. The fluidity of the membrane can also change depending on the ratio of saturated to unsaturated fatty acids in the phospholipids. So, for example, membranes that have more unsaturated fatty acids tend to be more fluid, as there's more space for these phospholipids to move around because they can't pack in tightly together. Whereas membranes that are high in saturated fatty acids are much less fluid because the fatty acids are stacked against each other, closely packed. This is important because cells need to be able to adjust to temperature changes. So, for example, to counteract the increasing fluidity of a membrane as the temperature rises, the cell's going to increase the percentage of the saturated fatty acids in its membrane. Cholesterol is also sometimes dubbed the fluidity buffer because it also plays a role in moderating fluidity and making sure it doesn't go too high or too low depending on the circumstances. So the other aspect that the phospholipid bilayer gives membranes is its selective permeability. So this is that idea where only certain things are allowed to cross the membrane into the cell where others aren't. So small hydrophobic molecules like oxygen and carbon dioxide, as well as a few uncharged small polar molecules, are able to cross the membrane relatively freely. However, large molecules like glucose, amino acids, things like that, as well as highly charged ions, will not be able to cross that hydrophobic core. So the question then should be asked, cells need glucose, they need ions, how in the world do these things get across the membrane? This brings us to the mosaic part of the fluid mosaic model. So just like a traditional mosaic shown above here, the membrane, if you look at it under an electron microscope, will give the appearance of a mosaic simply because the number of membrane proteins that are stuck in the membrane. So this now gives the membrane a bucket load of functions, which allow us to make connections to so many different concepts in biology. But before we get to those, let's just take a quick look at the basic membrane proteins that are found in the bilayer. So there are two different types. The first is called an integral membrane protein, shown in purple here. And these are any proteins that somehow penetrate the hydrophobic core of the membrane and thus are anchored in that hydrophobic core. So some of them only go partially in, as you will see in the first one there, whereas others will completely traverse the hydrophobic core and have hydrophilic portions on either side of the membrane. Sometimes they only pass through the core once, sometimes they go through multiple times, but ultimately that doesn't matter too much. The second one, the peripheral membrane proteins, are definitely associated with membranes, so they're still membrane proteins, but they're not embedded into the hydrophobic core at all, and rather they are held onto the membrane by interacting either with the phospholipid bilayer heads or with a hydrophilic portion of an integral membrane protein that happens to be sticking out of the membrane. And this is where we can start to make a whack of connections to different areas of biology. We talked a little bit earlier, asked the question, how do molecules get across the membrane when the phospholipid bilayer doesn't allow them? Well, that's where the transport membrane proteins come in. And there's a number of different types of these. So an example would be a channel protein shown here, which simply creates a hole in the membrane through which molecules can travel, usually down the concentration gradient. The second is what's called a protein pump. And this is a membrane protein that actively pumps molecules against the concentration gradient. And a good example of this is the sodium-potassium pump, which is involved in creating ion gradients across the membrane. And this is critical when it comes to the functioning of nerve cells, as you will see later on. There's another function of membrane proteins, and those are electron carriers. And these play a massive role in respiration and photosynthesis. So their job is to carry electrons from one protein to another inside the membrane. And we'll be talking a lot more about those later as well. Some integral membrane proteins also perform enzymatic functions. So few are as important as the ATP synthase complex shown here, which is in the business of making ATP in both photosynthesis and respiration. So down below here, we've got a lot of stuff going on, but one of the things that's happening is ATP is being synthesized from ADP and inorganic phosphate using the energy of electron movement and proton gradients. Some membrane proteins are also act as receptors for hormones and other molecules, and those are the first step in cell signaling pathways that allow different parts of the body to communicate with each other in response to different external or internal changes. So the example here shown is the epidermal growth factor that's binding to a receptor and starting a chain of events that is going to result in differential gene expression, and that will result in the division of cells. So a lot of things going on there. And we could go on and on and on, but hopefully this gives you not only the basic information on how membranes are put together, but more importantly, helps you understand that this relatively simple structure is involved directly in so many important aspects of biology. Thanks for watching.